Hi, I want to present quickly a very exciting study on multi-omics analysis of the response to Ritlecitinib, a novel JAK inhibitor in alopecia areata subtypes, and correlation with efficacy. So, alopecia areata is a disease that many of us know, but let's remember it's an autoimmune disease characterized by non-scarring hair loss of the scalp, but can be any body area. And Ritlecitinib is a novel oral selective dual inhibitor of JAK3 TEC family kinases that is approved now to treat severe alopecia areata in adults and adolescents over 12. And a biopsy substudy of the phase 2 Allegro clinical trial demonstrated that ritlecitinib not only improved clinically the hair regrowth, but also it improved the transcriptomic of patients with alopecia areata, so turned lesional scalp towards a non-lesional profile. And doing that, it also improved type 1 and type 2 immune genes and hair keratins, and there was some correlation with disease activity. Now, this study shows a post hoc analysis that examined changes in the molecular signatures in the lesional scalp of patients with different subtypes of alopecia areata that were in the study, both patients with more patchy hair loss and those with totalis and universalis. And we wanted to evaluate the differential responses to ritlecitinib and to evaluate correlations between potential biomarkers and the scalp hair regrowth. So this is the study design. And remember, the study enrolled patients with more than 50% scalp hair loss. So even those that we say have patchy alopecia areata, they still had substantial hair loss. And the patients here in this analysis were divided to patchy alopecia patients or those that have total scalp or total body hair loss, alopecia totalis and universalis. And just to orient everybody to the study design, it was a 24-week study. There were biopsies at baseline, lesional and non-lesional, and also week 24, and as well as week 12. There were also a few biopsies collected at week 4, but most of the biopsies were at baseline, week 24, and week 12. And we evaluated changes in lesional scalp, but also changes in serum proteins between pre and post. And of course, we did correlations between the changes in biomarkers and the clinical results. So let's review the data by this new division between patchy and totalis and universalis. So first of all, what do we see here? You see much more blue and much more red in the patients that have patchy alopecia. And why is that? Remember, the primary endpoint was at week 24. In alopecia, that's still very early. So what do we know? We know that totalis and universalis take longer time to grow hair, and that's probably why here you see that the results for totalis and universalis are not as striking as the results you have for patchy, where you have very dark red and dark blue, meaning you really modified the gene responses. So you see increases in hair keratins, and you see downregulation of type 1 and type 2 immune genes, and also genes that are associated with the mechanism of action of ritlecitinib. Now, in totalis and universalis, you see the same trends, but they are not as evident, and likely it's because it takes more time. If we would do it at 48 weeks, I bet the results will be similar, but it takes more time, and the reason being they have more systemic inflammation and they have the alopecia totalis and universalis. So remember, total body or scalp hair loss. So it will take more time, likely, to see the same type of inhibition of immune genes and increases in hair keratins. Now, importantly, we also wanted to correlate changes in the clinical scores by SALT and changes in biomarkers. And what do we see? We see something very interesting. We see that there is a correlation between baseline gene expression and scalp hair regrowth. So we see increases with hair keratins and we see decreases in immune genes. So you see that inverse correlation, and that is really important. So the more inflammation you have, it will take likely longer. And the same with hair keratin. So they go in different directions. And we also see that there is a correlation between lesional gene expression and changes in lesional gene expression and changes in serum protein levels. So pay attention that you have that correlation between hair growth and the level of biomarkers in the serum, as well as the changes in the level of biomarkers in the scalp. So we need to bring that into account when we have a patient in front of us that has totalis and universalis. There is a reason why it will take more time to grow their hair, right? They likely have more inflammation in their serum. They have more systemic immune abnormalities. And of course, that will relate to more inflammation that they have in the scalp, and that will take longer to reverse. So, to put it all together, treatment with Ritlecitinib improves the gene expression in lesional scalp and serum proteins in patients with alopecia areata and is correlated with hair growth regrowth response. And patients with more patchy forms of alopecia areata had greater response to Ritlecitinib than those with more global forms, the totalis and universalis, at earlier time points. And that reinforces the beneficial effect of treating early in the disease course. Let's not wait until they have totalis and universalis. We need to treat our patients earlier in the game. And I think that's very, very important. And 50% is just a number, but sometimes even less than 50% will necessitate a systemic treatment. And the biomarkers identified in this study play essential roles in the modulation of immune and cellular processes that are important for hair follicle health and thereby reducing inflammation and improving hair regrowth. And by identifying baseline molecular features associated with ritlecitinib treatment across the clinical subtypes, I believe this work supports the development of precision medicine strategies for patient stratification for patients with alopecia areata, but perhaps even beyond. Thank you so much, and I hope you enjoyed the paper.